Hi, this is Bio156, Chapter 10, Molecular Biology of a Gene. This is the first of 7 videos for this lecture topic. In this video, we'll discuss the structure of the genetic material, including how experiments showed that DNA is the genetic material, how DNA and RNA are polymers of nucleotides, and how DNA is a double-stranded helix. Chapter 10 focuses on molecular biology, which is the study of heredity at the molecular level. In other words, how we pass on information from one generation to the next using molecules. In molecular biology, as we study this topic, we are standing on the shoulders of giants. 100 years ago, we didn't know that DNA was the genetic material of life. And now we've come to the point where we can actually manipulate DNA in other organisms and even in ourselves sometimes. So the advances that we've made in our understanding and our abilities to manipulate or change DNA are astounding. Some of the scientists we'll talk about include Alfred Hershey and Martha Chase, who in 1952 helped to show that DNA was the genetic material. Alfred Hershey and Martha Chase were 2 U.S. scientists who were studying T2, which is a bacteriophage, or in other words, a virus that infects bacteria. T2 happens to infect E. coli, which are common bacteria found in our digestive system, which can sometimes cause disease in humans. What Alfred Hershey and Martha Chase were doing were they were trying to determine what the genetic material was. They knew that viruses, and this virus in particular actually, that this virus was made up of just protein and DNA. It had a protein structure or shell, and then it had DNA contained inside this capsule up here. And they knew that something was entering the bacteria cells and providing the genetic instructions for making more of these viruses, because the bacteria would split and release more viruses as they died. And so they knew that something from the virus was getting into the bacteria to direct this. And they knew that it had to be either protein or DNA because there was nothing else making up this virus. And so Alfred Hershey and Martha Chase used radioactive isotope labels. Remember we said earlier in Chapter 2 that we can trace radioactivity. So sometimes we use it as labels in scientific experiments. So they labeled sulfur in the protein, because sulfur is found just in protein and not in DNA. And they labeled the phosphorus in DNA, because phosphorus is more often found in DNA than in proteins. And then they infected E. coli with this T2 virus that had either radioactive sulfur or radioactive phosphorus. And then they detected where the radioactivity went. So here's the structure of that experiment. This is called the blender experiment sometimes, because they actually used a blender at one point in this experiment. And so Alfred Hershey and Martha Chase had 2 different setups. They had one batch of E. coli that they infected with virus that had the protein labeled. So the protein was radioactive, but the DNA was not. And they allowed enough time for the viruses to infect the bacteria. And then they put the entire thing, bacteria and viruses and all, in a blender, and literally just turned that blender on to shake things up so that anything that was left on the outside of the bacteria would be knocked off, and only things that had entered the bacteria would be detected with the rest of the bacteria. So then they centrifuged that slurry, that mix of bacteria and virus and liquid. When they centrifuged it, what I mean is that they put it in a machine that will spin it at very high speeds. And that separates things out by size. So the heavier things sink to the bottom of the tube, which is facing the outside of the centrifuge, and the lighter things stay in the liquid, which is in the center, which is towards the center rather. And so the pellet is what we call anything that's large and big and solid, and it winds up at the bottom of the centrifuge tube. And so that would contain the bacterial cells as well as anything that was inside the cells. And then the liquid would contain anything that had gotten knocked off of the cells. And they found that the radioactivity, when they were using radioactive protein, the radioactivity was in the liquid, whereas the pellet was not radioactive, suggesting that the protein was not getting into the cell. It was only the DNA getting in. And so then they did the reverse experiment to confirm this, where they labeled the DNA now and did the same thing. It allowed the virus to infect, blended it up, and then centrifuged it down. And they found now that the pellet was radioactive. So the pellet contained the bacterial cells and anything that had entered them, and the radioactivity was in the pellet, and they'd labeled the DNA with the radioactivity. So that means the DNA had gotten into the bacteria, and the DNA was what was getting passed on in order to code for more viruses to be made. So the infected cells had radioactive phosphorus that they had gotten from the DNA of the T2 and not from the protein. So infected cells had radioactive phosphorus that they got from the DNA of the T2 virus and not radioactive sulfur from the T2 protein. This evidence helped to demonstrate that DNA is the genetic material. It's what gets passed on from one generation to the next, or in this case from a virus to a bacteria, and it's what codes for more of that virus, or what allows more viruses to be made. So once scientists had established that DNA is the genetic material, other questions followed, like what is the structure of DNA, which was solved by Watson and Crick in 1953, a year later, and what is the underlying chemistry of DNA? To wrap up that experiment, let's ask a lecture quiz question. This is question 1 for the Chapter 10 lecture quiz. In an important experiment, radioactively labeled bacteriophages, viruses, were allowed to infect bacteria. In the first trial, the phages contained radioactive DNA and radioactivity was detected in the bacteria. Next, other phages containing radioactive protein were allowed to infect bacteria and radioactivity was not detected in the bacteria. These experiments led to the conclusion that A. The genetic material of this phage was made of RNA. B. The genetic material of this bacteriophage was made of DNA. C. Genes carry information for making proteins. D. DNA is made of nucleotides. Or E. Bacteriophages can infect bacteria. Go ahead and pause the video here while you write down your answer. Let's talk a little bit about the chemistry of DNA. You should remember most of this from our discussion in Chapter 3 about nucleic acids, and if you don't remember, I would suggest you go back and review that material. So, as you should recall, each DNA nucleotide, remember the building blocks are called nucleotides, and so we have long strands made up of many nucleotides linked together, but each DNA nucleotide has a nitrogenous base, which is this colored part that's sticking out here. That can be either A, adenine, C, cytosine, T, thymine, or G, guanine. Those are the 4 possibilities in DNA. And then that nitrogenous base is covalently bonded to a sugar, shown here in blue, which is also covalently bonded to a phosphate group, shown here in yellow. And the nucleotides are joined together by covalent bonds between the adjacent sugars and phosphate groups. So this sugar here is attached to that phosphate from the next nucleotide. and so on and so forth down the chain. So that creates a sugar phosphate backbone. So here's the structure of just a single nucleotide. It's got that nitrogenous base, that sugar, and that phosphate group. And this is in DNA, which stands for deoxyribonucleic acid. And it's called a nucleic acid because it's found in the nucleus. And it's an acid because of the phosphate group here, which can give off, or which can give up some hydrogens. These oxygens here originally were bonded to hydrogens, but they can give those up easily, increasing the hydrogen concentration of the solution, which makes it more acidic. So in this sugar, which is deoxyribose here in DNA, it has 5 carbons and just 2 oxygens, which is 1 less than the oxygens in ribose sugar. So that's what makes it deoxyribose. And then those nitrogenous bases have either a single or a double ring of nitrogen and carbon, and they have various different functional groups, like a methyl group here, carbonyl group here, an amino group right here. And those functional groups determine which nitrogenous bases can hydrogen bond to which other base. So for example, the amino group here can hydrogen bond with the carbonyl group here. And T-thymine will always bond with A-adenine and vice versa. A bonds with T, T bonds with A. And cytosine, or C, always bonds with guanine, or G, and vice versa. G always binds with C. Now, what about RNA? RNA has a very similar structure, but there are 3 differences compared to DNA. First, its sugar is ribose rather than deoxyribose. It has a 3rd oxygen here compared to the sugar in DNA. And number 2, the nitrogenous bases, instead of having T-thymine, we have uracil U. And finally, RNA is usually found just as a single strand on its own, whereas DNA is usually found as 2 strands hydrogen bonded together. Now let's talk about how that structure was determined. This is Chapter 10 Lecture Quiz, Question 2. Select all of the characteristics of DNA that are not shared with RNA. A, ribose sugar. B, adenine. C, cytosine. D, thymine. E, guanine. F, phosphate group. G, double-twisted strands. Or H, covalent bonds in sugar phosphate backbone. Go ahead and pause the video here while you write down your answers. So James Watson and Francis Crick were 2 scientists who in 1953 were able to figure out the structure of DNA. And they used the X-ray crystallography, this image here, that Rosalind Franklin had taken. Rosalind Franklin was a technician who was working in a lab close by at the time on X-ray crystallography, taking images of things using X-ray techniques. And they were able to determine, based on this image, they looked at this and they saw, oh look, you've got a darker spot, which represents a backbone of some sort on one side and a backbone on the other. And then there's an X in the center showing things coming through the center here, holding these 2 strands together. And we'll talk about the structure that they actually determined for DNA in just a second. And Watson and Crick won the Nobel Prize in 1962 for their work in determining the structure of DNA. Rosalind Franklin, unfortunately, had passed away previous to that. So, what they determined about DNA is that DNA forms a double-stranded helix. So a helix is like a loop or spiral. And if you have a double helix, that means you have two loops or spirals that are spiraling around each other. So if we untwist that spiral, we would be looking at what looks kind of like a rope ladder. So the sides of this rope ladder would represent the sugar phosphate backbone that we talked about, where the nucleotides are attached together. And the rungs would be the nitrogenous bases sticking out towards the center. And really, this wouldn't be a solid rung. There would be a space in the center because you have one nucleotide, one nitrogenous base, on each side that they're held together in the center by hydrogen bonds. I have a better diagram of this on the next slide to show you how that might look like. So we have that ladder, and then if you twist that ladder up, you would end up with the double helix structure of the overall DNA molecule. Now there is a nice animation of the DNA double helix. It's just about 1 minute long, so you can follow that link. That link is also up on D2L because I think the links on the PowerPoint slides don't really work for you guys. So go and find that one on D2L and follow that. Okay, so we mentioned how DNA is a double-stranded helix. So we have the sugar phosphate backbone represented by this blue and yellow ribbon on both sides that's wrapped around the helix here. And then we have the nitrogenous bases facing in. And those nitrogenous bases are hydrogen bonding together. If we zoom in on that and untwist it a bit, what we'd see is that the strands are actually running opposite each other. So you've got one strand where you've got sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, and then on the other side it's phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. And that orientation helps the hydrogen bonds to be oriented correctly so that the bases can actually bond together. And you can see those hydrogen bonds represented by dotted lines between the nucleotides. So between C and G, there's 3 places where hydrogen bonds are forming, and between A and T, there's only 2 places where hydrogen bonds are forming. So the 3 hydrogen bonds between G and C are stronger than the 2 hydrogen bonds between A and T. This is the Chapter 10 lecture quiz, question 3. DNA and RNA are polymers of what kind of monomers? A, carbohydrate, B, nucleic acid, C, nucleotide, or D, amino acid? Go ahead and pause the video here while you write down your answer. Right, so in review, DNA is the genetic material of life, and 2 strands of DNA wind together to form a double helix. Nucleotides, which are the building blocks of nucleic acids, are made up of a sugar, a phosphate group, and a nitrogenous base. And the nitrogenous bases in DNA hydrogen bond with the bases on the other side of the DNA strand to hold that molecule together. Let's try a practice question. The correct answer here is B, 30%. So if adenine, or A, makes up 20%, that means that T, or thymine, which it binds to, has to make up another 20%. So 20% and 20%, that would make up 40%. That means that 60% has to be made up of guanine, G, and cytosine, C, and each of them is going to make up half of that 60%, or in other words, each will make up 30%, B. Let's try one more practice question here. The correct answer here is D, hydrogen. So hydrogen bonds are what hold together the nitrogenous bases on the opposite sides of those DNA strands that are running opposite each other. And there is a word here that we didn't really talk about earlier, complementary. Complementary means that the strand on one side has nitrogenous bases that match or complement the bases on the other strand. They're not identical. Remember, we don't have A with A and T with T and so on. Instead, they're complementary. We have A with T and C with G and so on, so they complete each other. That concludes this part of the lecture. This has been Bio 156, Chapter 10, Video 1 of 7.